It's great to be here today among a bunch of phage lovers all in one room. My name is Mojgan Rabiey. I'm from University of Birmingham. And today I will talk about phage bacteria coevolution dynamic in cherry pathosystem. Just a brief introduction in cherry bacterial canker. So cherry trees are attacked by the pathovirus of Pseudomonas stringi causing canker. So there are three pathovirus of Pseudomonas stringi that attacks cherries. They are Pseudomonas stringi, pathovirus stringi, and Pseudomonas stringi, pathovirus morsporonorum, and there are two races. So I call them from PSS and PSM from now on. So not only there are three pathovirus of Pseudomonas causing this disease, but also there are different strains of each pathovirus out there that are affecting cherry trees. So cherry canker is an annual problem for cherry fruit industry in the UK and cause up to 75% loss of cherry trees. The Pseudomonas stringi pathovirus affects all parts of the cherry plant. So from, you can see on the leaves causing shot holes, on the fruit cause necrosis, and also on the trunk of the tree cause huge amount of oozing and bleeding. So it seems like that cherry trees bleed to death. So currently there is no cure for the disease because the traditional method of using copper compound is quite limited because of the laws and also because it's not effective anymore because of bacterial resistance. And also breeding for resistance is quite challenging because of the complex nature of the disease and because there are different pathovirus of Pseudomonas causing disease and each one has different pathogenicity and virulence. So an alternative method would be use of bacteriophages. So if we wanted to first see if we can find bacteriophages against different pathovirus of Pseudomonas stringi causing canker, and to understand how we can minimize the bacterial resistance emerging to phage, and also if we can develop phage cocktails that are most effective at reducing bacterial population in cherry. So we isolated 70 phages from different cherry orchards in the UK and from leaf, bark, and soil of cherry trees. The first thing we did, we host range assayed them against all pathovirus of Pseudomonas stringi causing canker and against all most strains of Pseudomonas stringi that we had. Also, we tested them against a range of beneficial bacteria to first test that they don't affect any of those bacteria, such as Pseudomonas fluorescens. So none of the phages we isolated could infect beneficial bacteria that suggested that it's safe to do more analysis on them. So based on the host range, then we narrowed them down to 13 phages and we genome sequenced them. So based on the genome sequencing, they belong to 5 different groups, and some of them being novel and reported for the first time. However, none of the phages carried any virulence genes in the genome, so suggested that quite safe to be used for further analysis. So the next step for us was to understand if phages can infect bacterial population in the lab and how effective they are. So also is what concentration of phage we could use that reduce bacterial population, but less bacterial resistant emerges. So I'm going to take you through the figures. So if you look at the MR2, so we measured optical density of bacterial population in the absence of phage and in the presence of phage at different multiplicity of infection. So on the top, you could see PSS grow exponentially throughout the 24 hours experiment. But the highest the concentration of the phage, let's see if I can, the highest the concentration of the phage, you could see the fastest the killing the bacteria population, however, more resistant bacteria emerges during 24 hours. But the lowest bacteria, the phage concentration we used, it takes the time to reduce bacteria population, but hardly any phage resistance appears during 24 hours experiment. So it was quite the same as for phage MR6 and other phages that we use individually. So now we know that which concentration of phage we could use, and then we move to the implant assay to understand how phages react in plant surfaces. So first we use the model plants in bean leaves. So we infected them with PSS without any phage, and also in the presence of each phage individually, and also a cocktail of phages or mixture of phages. Then we measured the colony forming unit of bacteria population every 24 hours for 96 hours. And if you look, so you can see in the top graph or top figure that PSS maintained its population throughout the experiment. But when we use phage MR6 individually, it reduced bacteria population quite significantly throughout the experiment. But when we use cocktail of 7 phages, that also MR6 is within that cocktail, it reduced bacteria population, but not as effectively as when we used the phage individually. So in another experiments we did on cherry leaves on a longer term period for 5 weeks. And then again, we infected the leaves with phages individually, also in a cocktail, that you could see phage MR6 on the top figure that reduced bacteria population more significantly than when phage used in a cocktail of those cocktail of 7 phages. Although we could see these fluctuations throughout the experiments, that it could be because of the arm race dynamic or fluctuation selection dynamic. But what's interesting for us here is to see why when we use each phage individually is more effective than when we use phage in cocktail or mixture of different of them. So then it led us to think it maybe phage compete with each other or antagonize each other, and that reducing the efficacy at reducing bacterial population. So we did another experiments when we used each phage from different groups based on their genome sequence. So we used each phage individually and also in mixture of 2, 3, 4, and 5 phages together. So we had like 31 different combinations of phages that we tested. Then again, we measured optical density of bacterial population in the absence and presence of phage during 48 hours period. So if you look at, again, PSS grows exponentially throughout the experiments, but when we use phage MR6 individually, it reduced bacterial population immediately, kept the population down. However, after 24 hours, you could see the resistance start to emerge. And you can see this bumps through during 24 hours to 48 hours period. But again, at the end, you can see the optical density is much lower when phage is present. But when we add a mix of 2 phages, MR6 and MR15, you could see the same pattern, kills bacterial population immediately, keep the population down, but after 24 hours, you can see the resistant bacteria emerge. But here you could see only 1 bomb on 1 arm race compared to the 2 we could see here. Same pattern for when we mix 3 phages, but now look here. If you mix 4 phages at the same time, after 24 hours, you can see bacterial resistant emerge, but we don't see any of those arm races that were present when phage MR6 or 2 of the phages were present. And the end optical density was much higher than when we use phage MR6 on its own. So if you look at the graph more closely, you could see that after 24 hours, we might see that bacterial resistant emerges, maybe phage evolution happened, then bacteria resisted again, phage evolution happened, and so on. But when we use mixture of 4 phages, we only could see bacteria resistant emerging, but no evolution of phages. So that must suggest that phages might antagonize each other or compete with each other at infecting PSS, and therefore it's affecting the efficacy at reducing bacterial population. So when we design a phage, it is important to understand which phages we mix with each other to reduce the population. So then it led us to think about this co-evolution of phage and bacteria together. So we wanted to understand this mechanism even better. So we conducted a co-evolutionary experiments that when we chose 1 phage from each group based on their genome sequences. So we have 5 phage that we mixed each phage with PSS individually, and then we kept the incubated that for 48 hours. And then we transferred the population of phage and bacteria to new broth. Then again, incubated for 48 hours. And we did this transfer for 10 times. And from every other transfer, we collected phage and bacteria population. And so that I call this B for bacteria and P for phage. So you know for the next slides. So then from this phage and bacteria that we isolated, we genome sequenced them and we looked at the SNP changes in the genome. So we analyzed the bacteria population for the cost of resistance to phage. So when we co-evolved this phage and bacteria, so here you could see this is a Pseudomonas stringi, Phages stringi that was co-evolved with MR6. And when we genome sequenced them, we saw mutations happening in the genome. And then also it was different when we co-evolved PSS and MR14. However, this glycosyl transferase was the mutation that was dominant in PSS MR6 interaction, and it was also present with PSS MR14 interaction, but also lipopolysaccharide was a dominant mutation here. So this glycosyl transferase is within the core genome or core cluster of lipopolysaccharide in PSS. And is also involved in lipopolysaccharide synthesis. And we know that lipopolysaccharide is the core component of outer membrane of Pseudomonas stringi or gram-negative bacteria. So if we look into the phage MR6 and its co-evolved generation, we wanted to see, okay, can it affect, can this co-evolved generation of MR6, can it still kill PSS? So yes, it's still, so it can. So this is again by looking at the optical density of bacteria population in the presence of all co-evolved phages during 24 hours. But we said, okay, can we now knock out this glycosyl transferase that is within the lipopolysaccharide genome and see what happens? Can MR6 and its co-evolved generation again reduce bacteria population? So we knocked out this gene and we see that the MR6 and its co-evolved generation can't reduce bacteria population anymore. So we did looked at the MR14 and its co-evolved generations to see if it can still kill PSS. Yes, it could. But when we knocked out the lipopolysaccharide kinase from the PSS and we created these mutants, the phage MR14 and co-evolved generations couldn't infect PSS anymore. So this would suggest that this lipopolysaccharide probably is a key receptor for MR phages, or maybe the MR phages are lipopolysaccharide dependent when it comes to the infection. So this would help us to understand how we could develop a phage cocktail, because if we use phages that use the same receptors to infect bacteria population, then would affect their that to compete with each other throughout the infection. Then we looked at the cost of resistance in bacteria population to MR phages. So we chose the PSS that was co-evolved with MR14, and we did a motility assay, like swimming and swarming. So motility is involved in pathogen movement, and it affects this pathogenicity. So we measured PSS that was co-evolved with MR14. We measured its swimming and swarming in the media in different time intervals. And we found that the motility of PSS population was not affected by coevolution. Also, we looked at the growth of those evolved PSS, and then again, the growth was quite similar to the ancestral or original PSS that we used. So it suggests that the motility and growth of PSS was not affected, and the mutation that happened in the lipopolysaccharide perhaps is the important part in this phage bacteria coevolution dynamic. So to conclude my talk is, so it is important to understand phage receptors in phage bacteria interaction. So that would help us to develop cocktails that we can precisely target bacterial pathogen, especially because this cherry pathogen is quite complex, different pathogens and different strains of each pathogen that causing disease. So important to realize that when we develop a phage cocktail. So there are some questions still to be answered, if now we created these mutants of lipopolysaccharide, can we find phages out there in cherry orchards that can infect those PSS with lipopolysaccharide mutants? And does this lipopolysaccharide mutation occur in planta? So if we do this coevolution in a cherry environment, does the same mutations happen? And how these mutations affect the pathogenicity of PSS. So is there any trade-off? So does the pathogen prefer to keep its pathogenicity and not resist to phage, or it prefers to resist to phage and lose its pathogenicity? And can we predict bacterial mutation in phage bacteria coevolution? Now we have a selection of Pseudomonas stringi pathovags from a pilot study that we did recently. So if we have a machine learning techniques, can we look at the genome and predict that these mutations happens in phage bacteria coevolution? So in that note, I just want to say thanks to all the collaborators in this project and lots of master and PhD students involved. And thanks for the funders, and thank you all for listening.
Hi, thanks for the nice talk. I'm Dibyendu from Exeter. I wanted to ask about when you said that when you had multiple phages together, all of these phages are having LPS as a receptor, and hence you do not see the arms race dynamics. But could it be that simply because there are so many of these phages, not enough mutant phages are being produced because all of them are competing? Hence, the arms race dynamic is not going on rather than actual antagonism of some other sort?
Well, it could be that, but the thing we observed here, so we know that each phage individually can reduce bacterial population, and we know that which concentration doesn't create that mutations or resistance happens. So what we were hoping to see that because each phage, when in the phage bacteria interaction, they should reduce bacterial population because when they are individually, they do. So when in the mixture, well, I guess that they all use this same receptors or lipopolysaccharide as their main target to attach to bacteria population. But when all of them are trying at once to use the same target, that they've competed with each other, so they didn't let each other to infect bacteria population, or one that dominated the population that started to infect bacteria population, but didn't let the others to do so. And so that's what we think.
Have you sequenced these strains to see if the host mutations are the same or different?
So we sequenced the one that we co-evolved together, but not in the mixture. So that perhaps would be the next step for us to use those mixture and see what happens in that PSS population.
Right. Thank you.
When you have bacteria that are resistant to one of your phages, are they sensitive to the other phages? Did you do those experiments? Just curious.
Yeah, so the PSS mutants, yeah. So yes, yeah. So we tested each those PSS mutants that emerges throughout the interaction, mostly also during the coevolution experiments, that we found that the latest generation of mutations that we created, the phages were most able to reduce bacterial population, or the mutants that appears. So that's why we think that when we co-evolve phages and bacteria, we could train or develop phages that are more effective and effective at reducing bacterial population. So that's maybe that is the future of developing phage cocktail, that look at the coevolution and find the phages that are more effective and use those to reduce bacterial population.
Well, thank you very much. That was a really interesting talk, both on the application, but also just a really good way of probing those dynamics. So thank you very much.